Hi friends, today we are going to discuss about pleiotropy. It's a type of gene interaction, pleiotropy. And the previous session we discussed about several other type of interactions like epistatic interactions, where we discussed about dominant epistasis, recessive epistasis, duplicate dominant, duplicate recessive, then inhibitory gene action, lethal alleles, and etc. etc. In this video, we are discussing about pleiotropy. Pleiotropy is nothing but, here, normally we know that one gene have one expression or one gene supporting or controlling the expression of a particular trait, one trait. For example, if we have a gene R means, it is responsible for flower color means there are two alleles, they are capital R and small r, and these two alleles are responsible for two phenotypic traits. Phenotypic traits means basically one trait with the two phenotypic expression. They are either capital R means red, capital R, small r means also red, while it is small r means it is white. That means one gene controlling one phenotype, isn't it? One gene is responsible for the production of a one phenotype. And in the case of epistasis, we saw another thing, that was here more than one gene controlling one phenotype. Right, in the case of comp pattern in chicken, there are two genes, they are together responsible, P and R, these together responsible for the production of particular type of phenotype, that is comp pattern. That's why because of the interaction of this kind of two genes, we will get four different kind of phenotypic patterns, right? They are rose, pea, walnut or single. And some other phenotypes are also there that we already discussed in the case in gene interactions, right? So, here but the condition is different. And one more thing, in the case of polygenic inheritance, we discussed different type of genes controlling single phenotype, right? There are 5 genes for controlling a single trait or 10 genes controlling a single trait, where we discussed how to find the number of genes involved and how we will find the contribution of a single gene and all, right? But here the scenario is different. Here we are discussing about a single gene which controlling more than one seemingly unrelated traits, unrelated phenotypes. Here there is only one gene, but the effect of one gene is multiple. The single gene can affect different phenotypes. Say for example, if this gene, in the previous case we told that this R gene is responsible for the trait flower color. But if this gene showing a pleiotropy means, this R gene alone, this R gene alone is responsible for, say example, flower color, say example, pod shape, another example, pod size or leaf color, likewise. That is, single gene has multiple effect, multiple phenotypes. Single gene is responsible for different phenotypes. Seemingly unrelated phenotypes are controlled by a single gene. So, because of this reason, if there is any mutation in this single gene has multiple effects. If there is a interaction, this is a mutation in this R gene, which will cause change in flower color, which will cause change in pod shape, which will cause pod size, change in pod size or leaf color. This is known as pleiotropy. Here, by definition, it is nothing but here one gene which is controlling more than one seemingly unrelated phenotypes. They are not related phenotypes, they are unrelated phenotypes. Here that particular gene is controlling more than one seemingly unrelated phenotypes. That is known as pleiotropy. The word pleiotropy comes from two words, pleion and tropos. Pleion means more and tropos means way, pleiotropy. The concept of pleiotropy was introduced by Gregor John Mendel. At the time of Mendelian period itself, there was some evidence about pleiotropy, but he was failed to explain why this is or what this is. Because in 1866, while studying in pea plant, Mendel observed that 3 traits in pea plant such as, this is the history. Now we are discussing about the history of this pleiotropy. Here when studied characters in, or when did hybridization in pea plant, Mendel observed that in pea plant 3 characters such as flower color, pod, seed coat color, flower color, seed coat color, seed coat color and axial spot. These 3 characters are showing a different kind of interaction pattern. These 3 traits, these 3 are 3 different traits. One is flower color, second is seed coat color and third is axial spot. But these characters showing a different kind of interaction pattern. They are inheriting together, and when there is a mutation in any one of this character, we can observe that the other two are also following the same mutation. Such kind of changes he observed. But he was failed to explain why this is. What is the phenomenon behind this? Later, lot of scientists, when this geneticists worked on this, and they finally found that these characters are actually controlled by a single gene. Since it is controlled by a single gene, any mutation in that gene will result in change in the phenotype of these traits. That is the reason for this kind of difference in interaction in these 3 traits. This is how that pleiotropy was introduced or was found. In 1910, the term pleiotropy, the word pleiotropy for this interaction, the term pleiotropy was given by Ludwig Plate in 1910. In 1910, Ludwig was the person who introduced this term pleiotropy, and he chose that term pleiotropy from 2 words, pleon and tropos. Pleon means nothing but the meaning is more, and tropos means way. More ways. There are a single gene can more ways to control a particular control different trait. That is the meaning behind this. Later, several works done on pleiotropic expressions, and Hans Grünberg, who actually put forward a mechanism for pleiotropy, and he divided the entire pleiotropy as 2, that is genuine pleiotropy and spurious pleiotropy. But later the work of Beadle and Chatham, one gene, one enzyme hypothesis and all, they all discarded this genuine pleiotropy concept. But the mechanism, the first description about the mechanism of pleiotropy was given by Hans Grünberg. But right now we are not following his mechanisms or the basics of his mechanism, but now we know that. We are explaining general universal pleiotropy in the terms of molecular methods. Now we know that how this pleiotropy, molecular basis of, what is the molecular basis of pleiotropy, or how this pleiotropy happens means, here we told that one gene is responsible for multiple phenotypes. How it happens means, it's nothing but one gene actually brought a cascade of reaction, and it will result in a different type of gene products, and this one gene is responsible for a cascade of reaction, and this, because of that, this gene is responsible for different traits. In the case of, while discussing about biosignaling, you might know about cascade of reactions. A single reaction is connected to some other reactions. Mutation, some steps. There are bifurcators molecules or proteins. When that protein activates, it is not only brought a one reaction but also have effect on some other pathways also. If there is a gene X or a gene product X, that is a protein X, it has multiple effect on different pathways. If the gene which is responsible for this X protein, then we can say that this X protein is responsible for these different pathways or different phenotypes. Why? Because that mutation in that X will result in mutations in these phenotypes or stoppage of that pathways. This is the actual mechanism of actually pleiotropy. So pleiotropy is why this pleiotropy or molecular basis of pleiotropy is nothing but one gene responsible for a cascade of reaction or one gene product will result in the starting or a bifurcator, act as a bifurcator or a integrator. It connect different pathways, a cascade of reaction. That gene product is acting as a bifurcator or that integrator or somehow it acts as it starts or it causes a cascade of pathways. That's why it is responsible for different phenotype. So this is all about the molecular mechanism of pleiotropy, and here we normally we divide pleiotropy into three types: gene pleiotropy, developmental pleiotropy and selectional pleiotropy. Gene pleiotropy, developmental pleiotropy and selectional pleiotropy. Gene pleiotropy, developmental pleiotropy, third one is selectional pleiotropy. That is, the classification is based on the effect of pleiotropy. Gene pleiotropy is nothing but here one gene product interact with the other gene product and results in a new phenotype, or this particular gene product has interaction with several other gene products, and mutation in that particular gene causes mutation in other pathways also. In that way it interacts with the many phenotypes, or it results in many phenotypes. That is known as gene pleiotropy. Developmental pleiotropy means nothing but that mutation in one gene will result in interaction or effect in the developmental pathway of organism, where it acts on the developmental cascade. That's why it results in different phenotypes. That is developmental pleiotropy. Selectional pleiotropy means nothing but a mutation or a pleiotropic change which results in better fitness. Say for example, a particular mutation or a pleiotropy gene undergoes a variation or mutation, a gene which is already pleiotropic undergo a variation. That mutation causes a better phenotype so that nature can select. But that will not persist for a long period. There might be change. That gene might change its expression by evolution, or by when it is time passes. Sometimes it may change its expression, or that mutation may cause us better fitness. This is known as selectional pleiotropy. So there are three types of pleiotropy based on their action or how it is come to the light. We can divide that pleiotropy into gene pleiotropy, developmental pleiotropy and selectional pleiotropy. Okay. So these all about the pleiotropy. Now it's time for see what are all the examples for pleiotropy. First example is phenylketonuria. We know that what is phenylketonuria. It's a disease condition where it is impairment in the phenylalanine metabolism. That is, phenylalanine hydroxylase enzyme is defective, and which causes that impairment, or that results in the defect in the metabolism of phenylalanine. Normally phenylalanine hydroxylase is the enzyme which is responsible for the metabolism of phenylalanine. How we got phenylalanine? Through our food. By our food we got phenylalanine amino acid, and we should convert that phenylalanine into or metabolize that phenylalanine. Otherwise it will cause the increase in the phenylalanine concentration in our system, our body. So that should convert into or metabolize that phenylalanine. That metabolism is made possible by an enzyme known as phenylalanine hydroxylase. That enzyme is coded in chromosome number 12. Gene present for that enzyme is coded in chromosome number 12. That if there is any mutation in that particular gene causes the impairment of phenylalanine metabolism, will result in the accumulation of phenylalanine, which will in turn causes different phenotypic effects such as That nerve system, it will affect the nervous system, nerve impairment, it will causes integumentary system failure and lots of that kind of phenotypic expression. So here a single gene which is responsible for phenylalanine hydroxylase in chromosome number 12. Mutation in that gene responsible for different type of phenotypic expression. Defect in your nervous system, defect in integumentary system. Right. This is known as, that's why we are including the phenylketonuria as a example for pleiotropy. One more example that we can see is sickle cell anemia. Sickle cell anemia, we know that the hemoglobin becomes sickle shaped. This is condition where hemoglobin becomes sickle shaped. Normally our hemoglobin is expressed as hemoglobin A. Sickle cell, the hemoglobin is expressed as hemoglobin H, HbH. And this is a mutation condition. How this mutation causes is, this due to a point mutation at the 6th position in the beta chain of hemoglobin, where there is a point mutation which causes changes to glutamic acid to valine. That hemoglobin consisting of beta chain and alpha chain that we know. In that one of the beta chain, at the 6th position, there is a point mutation which causes the conversion of glutamic acid to valine, which result in the formation of this type of a sickle-shaped hemoglobin. The sickle-shaped hemoglobin, when the hemoglobin becomes sickle-shaped, it is less able to carry oxygen. It can't carry enough number of oxygen, result in a series of phenotypic effects. Not a single effect, but it causes lots of effect. It is associated with a number of phenotypic effects. And it also results in one more phenomenon known as heterozygote advantage. What is heterozygote advantage means? It is actually a better fitness phenomenon where malarial parasites, those who are heterozygous for sickle cell anemia condition, are susceptible for malarial infection. Why? Because malarial parasite can't brood inside the sickle-shaped RBC. So that those who are heterozygous in sickle cell condition are less susceptible for or resistant to malarial infection. That is known as heterozygote advantage. It is also another phenomenon, another phenotypic advantage. So that means a single mutation or single gene mutation will result in a lots of phenotype. So it is one of the examples for pleiotropy. And several other examples are there. Albinism is another example. Marfan syndrome is another example. Schizophrenia, a typical type of schizophrenia, which is also another type of pleiotropy. There one gene responsible for different expression. In the case of schizophrenic condition, that particular gene responsible for this condition have two effects. If that gene is mutated in the childhood stage, means it will result in autism, and if that gene mutation happens at the earlier that adulthood stage, it will result in schizophrenia. That kind of variation is also given to the individuals or phenotype by the mutation of that particular gene. So this kind of interaction is known as pleiotropy. That means one gene which is responsible for different phenotype, expression of different phenotype. This is known as pleiotropy. So these all about the pleiotropy. In the upcoming videos, we will discuss about some other different kind of interesting gene interaction patterns. Okay.